Welcome back to physiology. We are now talking about muscles, so let's go ahead and just dive in. So when we think about muscles, we typically think about skeletal muscles first, and that's what we're going to talk about. So a couple things about skeletal muscles is skeletal muscles are voluntary, right? They're voluntary, they're striated. Each one has a nerve terminal, and there's these neuromuscular junctions. Now, that's important because that's how they contract. And they do something called excitation contraction coupling, and it talks about in terms of using ACH and troponin. So I think the best way to actually cover this is to do it on the board. So let's take it to the whiteboard and talk all the way through skeletal muscle contractions. Acetylcholine is released at the neuromuscular junction. Remember, that's important in skeletal muscle because they don't have those in smooth muscles, okay? Then that's what causes depolarization within the individual muscle fibers. Now, it's going to travel down the sarcolemma. What we need to know about the sarcolemma is that calcium is present in the sarcolemma. So when calcium comes in, it binds to troponin on those thin filaments, okay? The thin actin filaments. Remember, that's where tropomyosin and troponin proteins are present, okay? When calcium binds there, it causes a change in the tropomyosin that leads to an exposure of something called a myosin binding site. Okay? Those myosin heads are attached to ADP and are now able to bind actin filaments, and that's what forms something called a cross bridge across the thin filaments. The release of ADP causes myosin to pull on the actin filaments, causing the thin and thick filaments to slide past each other, and that's called a power stroke. So the other thing that these muscle fibers do, and you kind of heard about it when we were talking about on the whiteboard, but there's something called the sliding theory of contraction. And again, I think this is best if we work this out on the whiteboard too. So let's go ahead and take it there. So when a muscle contracts, the length of the thin filaments and thick filaments do not change. Instead, the sarcomere shorten. And when we talk about this in a picture form, a sarcomere is from Z line to Z line. That distance shortens. The other thing that shortens there is on this chart is I, all right? So the distance actually between the different myosin and the different actins, okay? That would be the H bands. Those shorten, but the actual myosin and actin sites do not shorten in length. So a couple things that we didn't talk about when we were up at the board was something called muscle spindles. Now, you can read through those paragraphs, but the thing I want you to know is that muscle spindles determine the length. So basically the rate and change of muscle length, and it's fine motor control. So these would be found in the eyes and in fine motor movements, okay? The other thing here is when we think about Golgi tendon organs, that's the force on a muscle, okay? So we got muscle spindles, length. I want you to think Golgi tendons, force, okay? Let's keep moving. Types of contractions, you guys can read through that. I want to spend just a minute on the different types of muscle fibers. Now, you've heard about fast twitch and slow twitch muscles. I want you to know a little bit about the difference. So when you have a slow twitch muscle or type 1, those have a greater resistance to fatigue. So those are ones that you can keep on going with, all right? They have slow myosin, but they have lots of capillaries, they have high myoglobin content, and they have numerous mitochondria, all right? So those are the slow twitch fibers. Now the fast twitch are the type 2, and they quickly fatigue, okay? They have fewer blood capillaries, lower myoglobin, and then fewer mitochondria. So I would just be aware, slow twitch type 1, fast twitch type 2, okay? Now, in terms of muscle fibers, you guys can read through that as well as muscle metabolism. I would just be a little bit aware of this. It's a little bit of biochem in there, but you can look at the oxygen supply and demand of muscles, okay? I'm going to move though to smooth muscles. Now, smooth muscles and skeletal muscles are different, right? The biggest difference is that smooth muscles do not have troponin, and their contraction is different too. Smooth muscles are also non-striated, as well as they have involuntary control. So a lot of it's controlled by your autonomic nervous system. And just like we took the skeletal muscle up to the whiteboard and worked it out with the contraction, let's go ahead and take the smooth muscle contraction to the whiteboard too, and let's get this worked out. So in a smooth muscle cell, this is what happens. Calcium comes in via ligand-gated calcium channel. And since there's no troponin, it binds to something called calmodulin. When that happens, it stimulates myosin light chain kinase. Myosin light chain kinase can then go and activate myosin, which then allows a cross bridge to happen, causing a contraction. What's so cool about smooth muscle as well is that if there's no phosphorylation of myosin light chain kinase and there's no energy, it can still bind to actin via what we call a latch bridge formation. No energy is needed there, and the muscle can still hold tension. So the last muscle that we need to talk about is just cardiac muscle. And cardiac muscle is interesting because it kind of falls into the skeletal muscle because it's striated and it has sarcomeres, as well as it has troponin and tropomyosin. But it's also similar to smooth muscles, and then it's regulated by the autonomic nervous system. A couple just different things too is that it also has a lot of gap junctions, which is great because remember how there's the SA and A node in the heart, which spreads the contractions. In terms of the heart too, they're said to be myogenic because of this rather than neurogenic like other muscles. So I think it's a good time to take a pause. Let's go ahead and answer a few questions. And when we come back, we are finishing up endocrine with the GI system, as well as the whole physiology chapter in general. So come back in a few minutes.